Okay, so the human is obviously a host to lots and lots of microorganisms, some good, some bad. So healthy individuals coexist with these microbes, and some of them can be pathogens, but they don't necessarily cause an infection. Others are not considered to be pathogens, and we just have a symbiotic relationship with them. Occasionally, however, something happens with the immune system or something, and the microbes are able to cause a disease. So this whole process of causing disease is a cycle. So we need to at least define disease because everybody hears it and you kind of think you know what it is, but if I asked you to define it, a lot of you would have a hard time actually putting that into words. So let's talk about that. So disease is technically the deviation from health. And there are many causes of disease, such as poor diet, genetics, aging, pathogens, and many more. And obviously, we're going to focus on the pathogens in this chapter primarily. So an infectious disease is the disruption of a tissue or organism caused by microbes or the products that the microbes produce. So the cycle goes like this: contact, which progresses to infection, which then ends in disease, and then just goes back to contact of another person or animal, and then infection and disease, and it just goes around in a circle. Okay, so let's define these terms because disease and infection, almost seem like the same thing. And I mean, I'll be truthful, I don't really understand the difference myself, and I use them sort of interchangeably all the time. But there is some subtle differences. An infection is the invasion of the body by pathogenic microorganisms and the resulting condition in the tissues, and that's from the World English Dictionary. Disease, from dictionary.com, is a disordered or incorrectly functioning organ, part, structure, or system of the body resulting from the effect of genetic or developmental errors, infection, poisons, nutritional deficiency or imbalance, toxicity, or unfavorable environmental factors. Illness, sickness, ailment would be other terms for disease. Another definition: any abnormal condition of a plant that interferes with its vital physiological processes. And you can kind of interchange plant for person technically because all of this is the same thing. But anyway, caused by pathogenic microorganisms, parasites, unfavorable environmental, genetic, or nutritional factors, etc. So an infection is caused by a microbe. A disease is not necessarily caused by a microbe, but could be. Okay, so not all contact leads to colonization. We're in contact with pathogens and other microorganisms every single day by the millions, but we don't get infected every single day, right? In other words, they don't colonize and create an infection or a disease. Even if they do colonize, they don't necessarily lead to an infection. So even if you begin getting a colony of bacteria or something, it doesn't necessarily make you sick, right? And then not all infections lead to disease. So it can stop at any point in this continuum. So contamination without colonization and colonization without disease are the rule. In other words, far more often we have contact and colonization with infection that never leads to disease than does. So we've talked about this a lot. Last chapter we spent some time on it, but it's this concept of resident biota or normal flora or normal biota or indigenous flora. It's the normal microbes that live in or on our body that we live in some sort of a symbiotic relationship with. Okay, so it's bacteria, fungi, protozoa, viruses, and even arthropods, like those little tiny little mites or whatever they're called that live on our body and live on like dead skin cells and stuff. Um, the Human Microbiome Project, sort of like the Human Genome Project, where they're mapping out all of the genes. With the Human Microbiome Project, they're using metagenomics to identify the microbial profile in humans. In other words, they're taking all of the DNA sequences that are found from all of the various organisms living on or in the body and mapping them out to see what is considered to be normal for a person. The reason why is because they can't necessarily culture all of these things in a lab to be able to determine which one of these are living on us or not. So they're just, essentially scraping off samples from all over the body and just PCRing them and looking and mapping out the genome to see what's supposed to be there. So once this is finished, it's going to determine the normal biota that is in the human, and also the ones that aren't normal, so that they can determine what might be causing a disease. So how do we get this normal biota? There on or in the body is a huge variety of microbial niches or niches. It's some sort of French word that just means place to live, basically, or place to be. In other words, there's lots of little nooks and crannies and fluids and whatnot for these microbes to live in, and they're so varying that we're going to get a varying, I guess, variety of microbes living in them, right? So most areas of our body that have contact with the environment have some sort of microorganisms living in them. I don't know which ones don't. Maybe just like the eyes, potentially, because of the enzymes and white blood cells and whatnot that attack the organisms that enter. But I don't know where else. Um, Now, internal organs and tissues and their fluids normally microbe-free. Normally, occasionally there's going to be some microbe in the blood that may not necessarily be causing a disease, but typically this is considered to be microbe-free. The majority of microbes that come into contact with the body are destroyed by our immune system, our lymphocytes, and our other white blood cells before they ever have a chance to colonize. So the microbes that remain are in relationship with the human so that they're not necessarily harmful to the host. They're either commensal, which is where the bacteria benefits, but it does not harm the host, or they're mutualistic, which you remember from a previous chapter, means that both the host and the bacteria or the microbe both benefit, right? So they help each other out. So there's this concept called microbial antagonism. So an antagonist is something that goes against, right? So this is the antagonistic effect of our normal flora on the pathogens. In other words, they compete with each other. So the normal flora are not likely to be displaced when microbes come in, which means that because there's a limited number of attachment sites and there's already normal flora there, it's less likely that a pathogen is going to attach there and begin to colonize, right? So that's just competition for these niches or niches. Another reason this exists is because of the environment that the actual normal flora create. For example, in the vagina, the normal flora produces acidity, and that acidity makes it unlikely for microbes to colonize, pathogenic microbes, I should say. So our normal flora is beneficial, or at worst commensal, to a person who is in good health. Now we will talk about a type of an infection, and we may have already, I don't remember, where a host does become infected with the normal flora. But in a normal immune system, in a normal host, that won't happen. So, like I just said, in a compromised immune system, it potentially could create a disease. So there are factors that make the host defenses weaker. So if you have weaker host defenses, you're obviously going to be more susceptible to having a disease, right? For example, old age, because during old age, everything slows down. Nothing functions as good as it did when we were younger. But extreme youth, such as infancy or prematurity, um, is a problem, but for a completely different reason. It's because we haven't been exposed enough to the pathogens to create the host defenses against them, and we'll talk about that in like chapters 12 and 13. Genetic defects in immunity, as well as acquired defects in immunity, such as during the AIDS virus. Surgery and organ transplants, because when we have an organ transplanted, we have to go on an immune suppressing drug. Even though they try to find a match isn't ever a perfect match. The only way it would be is to have your own organ transplanted in yourself, but they have not perfected that yet. They're in the process of growing organs in the lab, and then someday they probably will be able to grow an organ from your own stem cells. And in those cases, you wouldn't need to take the immune suppressing drugs, but until that happens, you've got to take some immunosuppressant. And because of that, it makes your immune system not function properly. Therefore, you're more likely to get some sort of an infection. And then other underlying disease, such as cancer, liver malfunction, diabetes. So those things already put such a tax on your body that it is more likely to be infected. Chemotherapy from usually cancer, and then immunosuppressive drugs I already mentioned for organ transplants, but you also might take an immunosuppressive drug for something like rheumatoid arthritis or lupus, because those are autoimmune disorders, right? Physical and mental stress is are both common ones. Have you ever seen, you have a stressful week or something, and then at the end you get sick? It seems to happen more often than it probably should, so there may be some truth to that. Pregnancy, for whatever reason, I don't know, or other infections. So this is just a table that shows the site of our normal flora. So skin, respiratory tract, GI tract, like E. coli, what Lactobacillus acidophilus, bifidobacterium, stuff like that. Outer opening of the urethra, external genitalia, inside the vagina, and so on. Well, and apparently the external eye. So I don't know where I said earlier that maybe that was the one place we didn't have flora or normal flora, but apparently we do. So I don't know where. Anyway, it doesn't really matter. This table, anatomical sites and fluids thought to be sterile. So basically everything inside of your body that's closed to the environment. So if it's a body cavity that is not open to the environment, then it should be sterile, such as your lung cavity. Now that's not the airways of your lungs. That's around your lungs, in your thoracic cavity, in your abdominal cavity. So the cavities such as inside your GI tract, those are open to the environment. Those are not sterile. Internal eye, I'm not sure about sinuses. They communicate with the nasal cavity, or the middle ear in the case of the mastoid sinuses. Normally they are going to be sterile. Certainly less often though than the other cavities. Blood, urine is, as long as it's in the bladder or above, is sterile. Once it hits the urethra, it's no longer sterile. Cerebrospinal fluid, so on and so forth. So inside of the uterus should be sterile, and certainly inside of the amniotic sac is sterile. So an infant, or I should say a fetus, develops without any kind of normal flora. But obviously at some point they have to get introduced to these bacteria and other organisms. So how does that happen? Well, first starts when the fetal membranes are broken. which people call the water breaking. And so that breaks the barrier between the outside and the fetus. So premature rupturing, sometimes a lady's water will break, a month before the baby is actually born, and that can pose some risks for the fetus. And so sometimes they'll give the mother antibiotics. So in general, though, the exposure to these normal flora occurs during the actual delivery process. So the first area is just the normal flora that lives in the vagina. So what we call the birth canal. So as the baby passes through, it's exposed to huge numbers of bacteria and fungi and whatnot. So one implication of that is when a baby is born with cesarean section, it's actually not colonized with the same bacteria. It's colonized more likely with skin bacteria or organisms. So I guess biota, right? Like staph and things like that. So either way, staph, strep, lactobacilli are going to begin to colonize, but different strains are on the skin than are in the vagina. And it is generally considered to be healthier from a normal flora standpoint to have a vaginal birth as opposed to a cesarean birth. So we also, besides the bacteria living on the now infant, the intestine also has to be colonized, right? So it's mainly we're talking about the colon, but there should be bacteria in the entire GI tract to some degree. So bottle-fed infants have a mixed population of coliforms, which if you know anything about like sewage treatment and water purification, coliforms are what they test for to determine the safety of water. But lactobacilli, enteric strep, staphylococci. But with breastfed infants, bifidobacterium is going to be much more likely to be present. And these are often, if you supplement with a probiotic supplement, this is probably the most common one, if not, or it's certainly common, if not the most common. So these things metabolize the sugars in the milk and turn them into acids, and that's one of the things that helps compete with the pathogens. So that's one of the reasons why it's considered to be healthy to have normal flora. And then, of course, just as a person lives, they're going to be constantly exposed to different types of flora. Which of the following body sites is not colonized by known normal biota? Skin and mucous membranes, external genitalia, GI tract, kidneys and bladder, respiratory tract, and normally you should not have bacteria inside of your kidneys or inside of your bladder. So what is a pathogen? We say it all the time. The actual definition is a microbe whose relationship with its host is parasitic and results in infection and disease. And the pathogenicity is essentially just how effective a pathogen is, basically. Now, in your book, there is a table, or a figure, I guess, 11.2. And what it's trying to illustrate is, and it's using these sliders, like a fader on a soundboard or something. These don't exist, obviously, in our body. But what it's trying to show is just to use a comparison to show you why a person can get infected in the circumstances that need to be present for that to occur. So this over here is the microbe, and this is the host. So if a microbe has a low virulence, it's less likely to cause an infection, right? Also, there's something that we either already talked, actually we're going to talk about it a little bit later, called the infectious dose. If the infectious, and that's just the amount of bacterium or whatever that has to be present to actually cause infection. So if the infectious dose is low, then of course it's going to also be less likely to cause an infection. Another thing is you might be exposed to some sort of a pathogen, but if it's not in the right place, for example, if you've got a, I don't know, something that's transmitted orally, but it just touches your skin, you're not going to get an infection. So if the portal of entry is incorrect, then you're not going to get infected. Now, on the other hand, over here, if you have the host, so this right here represents the genetic profile, and so genetics plays an important role in whether or not a person is going to get in, or any animal is going to get infected, for that matter. For example, this is probably the most extreme, but there are viruses or bacteria that will infect, for example, a dog that won't infect a human, because we don't have the same attachment sites or the same types of compounds or whatever the case may be. So just genetically, we just aren't a match. Okay, so if we have a high resistance, then we're less likely to get an infection. If we've had a previous exposure, then our B cells, our plasma cells, which are a type of a lymphocyte, are creating antibodies, and so we are going to be able to combat the exposure much more quickly, and so we're less likely to get an actual infection. And then, of course, just the general health level. So essentially, if we have all of these sliders and switches in these positions, then the microbe is going to pass through and, or not necessarily pass through, but essentially not cause an infection. So in other words, you don't get an infection if all of these circumstances are this way. But let's look at some other circumstances. So what if we have a higher virulence of some sort of microbe, and we have a higher infectious dose, and they get into the correct portal of entry, but we didn't change these sliders over on this side, or the switch, which means we're still high resistance. We still have had previous exposure, which means we've created antibodies, and we're still a healthy individual. So it still could pass through without any kind of an infection, or it might begin to colonize, but not necessarily go into a full-blown infection or create the disease, right? So how about another scenario? What about where we have a little bit lower virulence, very low infectious dose? We didn't get the correct portal of entry, still have a high genetic profile that resists the disease, but we've never been exposed to it before. So this is the first time we're being exposed to this particular microbe. So it may pass through unnoticed because of the not being in the correct portal of entry, but it also may cause an infection or colonization, but not necessarily lead into full-blown disease. So what this is trying to illustrate is there are varying levels of all of this. So how do we end up with a disease? Well, the most likely scenario is this. We have something with a high virulence. We have a high infectious dose, which means we're exposed to a lot of it. It gets into the correct portal of entry. We have a lower genetic resistance. There are some diseases that we have zero genetic resistance for example. We've never been exposed to it before, so we've never created any kind of antibodies, and we may not have as high, just a general overall health. And so this is where we're more likely to actually have a full-blown infection. So the pathogenicity, as I said, is the organism's potential to cause an infection or a disease. So true pathogens are capable of causing a disease pretty much all of the time in a normal, healthy person with normal immunity. I shouldn't have said the word immunity because that implies that you are immune to something. So just normal immune defenses being present. So we have an active immune system. And they're generally associated with a specific recognizable disease like tuberculosis. So they may vary in severity from mild to severe or even fatal. And examples, the flu, the plague, malaria. So opportunistic pathogens are these pathogens that cause disease when the host's defenses are compromised. And they can become established in a part of the body that's not natural to them. So these are actually normal flora. They're not considered to be pathogenic. Well, not necessarily normal flora, but not necessarily pathogenic. So in other words, it can be something that you could be exposed to normally and have no issues. Yet if you have compromised immunity, then that thing that would normally not cause any kind of a disease could now potentially cause a disease. So the reason why they're not normally pathogenic is because they have low virulence. So an example is some species of Pseudomonas, which is a genus, as well as Candida albicans, which is yeast. Pseudomonas is a gram-negative aerobic, actually. Bacterium, there's like 200-ish species. So, and normally they don't cause any kind of issues, and they're fairly ubiquitous. Virulence is not the same as pathogenicity, and I know they seem the same, and they're also used interchangeably oftentimes, although not necessarily correctly. But virulence is the ability of a microbe to establish itself in a host and cause damage. And you could also throw into this also its resistance to being killed by our immune system too. So a virulence factor is anything that essentially helps it to survive or create more damage. So we mentioned earlier portals of entry. So as part of this becoming established in the host, we have to have the right portal of entry, right? And this is just the route that a pathogen will normally take to cause an infection. So there's these two words, exogenous and endogenous. And an exogenous microbe is a microbe that originates from a source outside of the body. Endogenous is something that's in or on the body. So most pathogens have a specific portal of entry, such as oral or through a lesion of some sort or something like that. So for some of these pathogens, if they get introduced in the wrong portal of entry, they won't ever create an infection. So for example, if the flu virus is inoculated into the nasal mucosa, it will create an infection, or could. But if it's in contact with the skin, that's the incorrect portal of entry, and there's no infection that's going to occur. Now, sometimes certain infectious agents can enter more than one portal of entry, like Mycobacterium tuberculosis. Its portals of entry are the respiratory or the GI tract. Certain strep and staph species can either enter through the skin, the urogenital tract, or the genitourinary tract, sometimes it's called, or the respiratory tract. So this table right here just lists certain portals of entry and some common organisms that are involved in them. So some interesting ones. Certain worms can burrow through the skin. Oftentimes you'll be exposed to those like in water. Some of these, like strep and staph, that actually normally will live on the skin, cause no issues until you get some sort of a cut or a break in the skin. Herpes simplex, which causes cold sores, if it's exposed to the mucous membranes, can cause an infection. A lot of these from insect bites, West Nile virus, that's from mosquitoes, same with malaria. A whole slew of them are inhalation. So when these airborne bacteria or viruses enter into your respiratory tract, they can cause infection. Another common way is through the urogenital tract. These would be, like all these bottom ones here, are all sexually transmitted disease, STDs. And then, of course, a whole host of foodborne illnesses would be through the GI tract, right? So the size of the inoculum, which is the size of whatever is inoculating, a person. We call this the infectious dose, or the ID. And this is defined as the minimum number of microbes necessary to cause an infection. So if it's below this threshold, it will not cause an infection. So microbes with a smaller infectious dose have a greater virulence. For example, the infectious dose for rickettsia is one single cell, which means you get exposed to one cell of rickettsia and you are going to get sick. And rickettsia causes typhus or spotted fever. The infectious dose for tuberculosis, giardiasis, coccidiomycosis is about 10 cells. So giardiasis is from Giardia lamblia. Tuberculosis is TB. That's generally a respiratory disease, but also can cause lesions in the spine and some other issues. Infectious dose for gonorrhea is about 1,000 cells. Infectious dose for typhoid fever is 10,000 cells. Cholera is 1 billion cells. So the point of this is just showing that there's a very wide range of infectious dose, and this just has to do primarily with the virulence of whatever is being causing the infection. Okay, so step 2 is actually becoming established in the host, and that requires something called adhesion, which is where the pathogen obviously attaches to the host. And this is dependent on the specific molecules between the host and the pathogen, and that's part of that genetic component, is if not all animals have the same types of attachments. Therefore, if they're absent, then it's obviously not going to be able to adhere. So certain organisms it can't bind to. So almost always a prerequisite for causing disease, because we all have different mechanisms for getting these things out of our body. So if we can just flush it out, it's never going to have a chance to attach. So the mechanisms, there's a whole bunch of different ways they attach. Some bacteria have fimbriae or pili, which are extensions off of the surface of them. Certain surface proteins that will attach to other proteins. Adhesive slimes are capsules like dental caries. Viruses can attach with specialized receptors. Parasitic worms have suckers or hooks or barbs that they'll actually embed into like the intestinal wall. So once they have become established, then they have to be able to survive the defenses of the host, right? Because we're immediately going to begin to start attacking them. So our normal flora don't get attacked, but if they are not normal flora, they're going to immediately begin to be attacked by our immune system. One of the cells that will do this are these cells called phagocytes. And so phagocytes are cells that engulf and destroy them. They're either going to be white blood cells or lymphocytes, or they'll begin their life as one of those things and end up in a tissue such as a like Kupffer cell or a dendritic cell in the skin, for example. So once they engulf them, I'm sure we've talked about this, but they then release lysozymes and some other things into the vacuole and it destroys the pathogen. There are also some antiphagocytic factors, though, that are produced by some of these bacteria that make it so they don't get phagocytized or engulfed or eaten. Okay, one of these groups are called leukocidins. Leuc means white, cide means kill. And so these things are produced by strep and staph species, certain ones, and these actually kill the phagocytes. So leuc is implying a white blood cell. That's why it's leuc means white. So they kill white is literally what that means. Another antiphagocytic factor is a slime layer or a capsule. And this just makes them tougher or makes it so that the actual phagocyte can't even get to them in the first place. And so Strep pneumoniae, Salmonella typhi, Neisseria meningitidis, Cryptococcus neoformans, these all will produce either a slime layer or a capsule. So another interesting thing is some bacteria can actually live inside the phagocyte. So the phagocyte engulfs the thing, and then instead of it killing it, it actually lives inside of it. So Legionella, Mycobacterium, and some Rickettsias can do this. So assuming it survives the host defenses, then at that point it's going to go ahead and flourish, right? And that's when we actually cause the disease. So we had mentioned what virulence factors were a little bit earlier, but let's talk about how they actually contribute to the pathogenicity. So there's three ways that these microorganisms are going to damage the host. Either directly through enzymes that are released, or directly through toxins that are released, both endotoxins and exotoxins, and we'll talk about what those are in a minute. Or indirectly by inducing the host's own defenses to become overactive or act inappropriately and do things rather than helping, they actually cause harm to the host. So let's go through each one of those, starting with enzymes. So these enzymes that are produced by these microbes are actually called exoenzymes. And they do things like break down tissues to obviously create damage, right? They can also dissolve the host's defense barriers, and what that does is instead of the diseases being compartmentalized, it allows them to spread to a larger area. So we have natural compartments built into us, and we also will create compartments in the form of like an abscess. But these enzymes are always fighting to break those apart to actually be able to spread. So examples of some of these enzymes: mucinase. Sounds like a over-the-counter cold medicine, right? But it's not. It's an enzyme, and this digests the mucous membrane. Hyaluronidase. There's something called hyaluronic acid, which is a component of our connective tissue and cartilage, and it makes up the ground substance, which is just the base substance that holds this all together. It's part of what makes that up, and so if it digests that, it essentially turns your tissues into jello, basically, right? Coagulase causes clotting of the blood or plasma. So it's a coagulant, right? Which a coagulant causes a clot to form. Kinase does the opposite. It actually dissolves the clots. We actually produce that naturally, but when it's produced unnaturally, it makes it so a person just bleeds and bleeds and bleeds. So toxins are the next category. So these are chemicals produced by other organisms that are poisonous. And they name these toxins according to their target. For example, a neurotoxin is toxic to the nervous system. Enterotoxins. Enter means intestine, right? So enterotoxins cause reaction in the intestines. This would be like a food poisoning. Hemotoxins, lyse red blood cells. Heme means blood. Nephrotoxins, nephro means kidney. So these things create damage to the kidneys. I mean, you could just go on down the list. Hepatotoxins would be liver and so on, right? So there's two categories of toxins. There's exotoxins. Exo means outside. And these are proteins that are produced by the microbe that have a high specificity for some target cell. So very powerful, sometimes cause cell death. And the reason why they damage the cells is it varies. It's either because of damage to the cell membrane, which causes lysis or explosion or whatever of the cell, or they could disrupt the functions inside of the cell, just mess up the machine and make it so it no longer functions. So an example of an exotoxin are these things called hemolysins, which disrupt the red blood cell membrane and cause it to fall apart and release hemoglobin. And these things are released to increase the pathogenicity of Streptococcus pyogenes. The enzyme that they produce or not the enzyme, but the toxin they produce, they're called streptolysins. Staphylococcus aureus produces alpha and beta toxins that cause hemolysis. So when you plate these things in blood agar, what you see are these zones of hemolysis, and that's where there is no red blood cells, because the bacteria digested the blood. And so that's what you're seeing on this picture over here. So here's the normal blood agar. So where it's white or green over here, that's the zone of hemolysis. Endotoxin is the lipopolysaccharide portion of the outer membrane of gram-negative cell walls. So what happens is, as these bacteria die, they actually shed their lipopolysaccharide into our circulation, and our body recognizes that, and it causes some specific things to occur. Systemically, meaning it's throughout the whole body, right? One of the things is fever, inflammation. It can cause hemorrhage, which would be local. Also diarrhea, as your body attempts to try to get rid of whatever's going on. So some examples of microbes that do this: Salmonella, Shigella, Neisseria meningitidis, E. coli. All of these things will release endotoxin, and it can lead to something called fatal endotoxic shock, which is obviously deadly. That's why it's called fatal. So more often than causing a direct damage is causing indirect damage. And this is due to excessive or an inappropriate immune response. So your immune system just goes nuts, basically. So this just compares the exotoxin with the endotoxin. So some things to kind of point out here. Exotoxins don't need a whole lot. You need to have high doses of endotoxin. The exotoxins are more specific to cell type, where endotoxins are systemic and cause systemic reactions like fever and inflammation. Endotoxins, as we mentioned, are lipopolysaccharides, and exotoxins are proteins. Exotoxins, denatured by heat. This is probably maybe partially the reason why we have fever, although 60 degrees is not even anywhere near our what our temperature is when we have a fever, but potentially helps. I don't know. Exotoxins will stimulate antitoxins to be produced by our immune system. Endotoxins don't. Exotoxins usually don't create a fever. Endotoxins usually do. And exotoxins are released from living cells. They're producing it and excreting it. Endotoxin is released when the cell actually dies and falls apart. And with endotoxin, it's always gram-negative bacteria, and exotoxin can be either one. So this is just this table here is just trying to illustrate, is trying to illustrate these different methods that we just talked about. So this is illustrating enzymes. And so what it's trying to show here is that these bacteria, when they release these little enzymes here, it causes the cells to disrupt. So it's actually breaking the connection between these cells, and then that allows obviously the barrier to be broken, and it allows these bacteria to spread. Toxins broken up into either exotoxins and endotoxins. This is showing tetanus, so you step on a nail. Clostridium tetani enters into your bloodstream, it begins to grow, and it releases the toxin tetanospasmin, which causes you to get what they call tetany, which is like lockjaw is a form of it, where your muscles contract and you can't release them. And then this over here is illustrating endotoxin. The cell falls apart. This pink stuff here, which is the lipopolysaccharide, is released throughout the blood, and then you end up with like a fever. This one here, as I said, is the most common. And the example that they're using here is Streptococcus pneumoniae because it's showing pneumonia. So what happens is it causes the body to release too much mucus, and it fills the lungs up with mucus. And then obviously you can't breathe. And the other thing that does is it gives a really nice place for bacteria to live. So there are all kinds of types of infections. And this table is trying to illustrate some of that. So let me just point out a few things here. So a localized infection, is just as it implies, is confined to a specific area. So if you get a cut, for example, and you end up with gangrene, you have a localized infection. You don't have gangrene throughout your whole body, right? A systemic infection means that it is throughout your whole body. So or at least several sites. So it travels usually through the bloodstream all over the body. So examples of that, chickenpox. That's a pretty simple one because almost all of us have probably had that. A focal infection, I'm not exactly sure how this is different from a localized infection, but streptococcal pharyngitis, which would be like strep throat, would enter into your mouth, for example, and infect your pharynx and not travel or cause any other infection anywhere else. A mixed infection would be an infection with several different bacteria all infecting us and causing damage at the same time. This term primary infection, this is the initial infection, right? So obviously implying that there's a secondary infection, which would be a second infection from a different microbe. So you get another infection on top of the first infection. The term acute infection comes on rapidly and is short-lived. Example of that is flu. Only lasts 7 to 10 days unless, of course, you die. Chronic infection would be something that progresses slowly and persists over a long period of time. For example, HIV is, at least for now, your whole life. However, I did just hear, I don't know if this is a rumor or not, but I did just hear that there has been somebody who has been cured of HIV. I don't know if that's true or not. I didn't research it, I just heard it. Okay, so the process of infection and disease, establishment, spread, and the pathologic effects. So one of the ways disease is caused is by obstruction of tubular structures like blood vessels, lymphatic vessels, fallopian tubes, bile ducts. And what it can actually lead to is something called necrosis, which is tissue death because of accumulated damage due to pathogens. It could be hypoxia, for example, because the blood can't flow to the area, or other issues. But whatever it is, it causes the cells to actually die. Viruses can cause destruction of the cells by actually lysing them, because remember they accumulate inside of the cell, and then once the cell lyses, then the viruses are released and they can go ahead and infect another cell at that point. So signs and symptoms, you hear this all the time, and you need to understand. In fact, you guys probably already know because you're of the program that you're in, but signs and symptoms are not the same thing. So just in case you don't know, let's go over this. So a sign is objective findings. Those are things that an observer can measure and see on their own without having to be told what it is. A symptom is subjective. So this is something that the patient feels or senses somehow, but cannot necessarily be seen by an observer. A syndrome is a disease that's identified by a certain complex of signs and symptoms. So this, an example, isn't a syndrome caused by a pathogen, but everybody's heard of it, so I'm just going to use it, like Down syndrome. Down syndrome can be recognized based on observation of the person who has Down syndrome, such as a simian crease in the hand, shape of the eyes, shape of the head, and enlargement of the tongue, and things like that. So inflammation is a response that our body has, and it has some specific symptoms sometimes, such as, and these don't always all have to be present, okay? But here's some that can be present: fever, pain, soreness, swelling. I don't really know why this is listed under symptoms, because all of these, well, it's not true. Fever and swelling, at least, both can be objective signs as well. But the signs are edema, which is swelling, granulomas and abscesses, which are when your body tries to compartmentalize an infection, sometimes it'll create an abscess. And so you get just like a pocket of pus, basically, which is just white blood cells and microbes all fighting each other. Lymphadenitis, which is swollen lymph nodes, which is really a misnomer because adene means gland and lymph nodes aren't glands. What we're talking about here is, have you ever been to the doctor and he, with some sort of a cold or flu or something, and he or she feels underneath your chin or neck area to feel for enlarged lymph nodes? Oftentimes people call these things swollen glands, which is not true at all. They're not glands, but that's what we're talking about. So your lymph nodes will actually swell in response to an infection, and that's actually normal. So a blood infection is particularly serious, and there are different kinds you can have. So one of these is called leukocytosis, and this is when you get an increase in white blood cells. Leukopenia is the opposite. That's a decrease in white blood cells. Leuc means white, cyte means cell, right? Penia means decrease. Septicemia, and heme means blood, like heme, is when we have microorganisms that are multiplying in the blood. which means they're present in large numbers. Bacteremia or viremia is the presence of bacteria or viruses in the blood, but not necessarily multiplying. So when you're septic, that means you have essentially a blood infection. So sometimes we get an infection, and someone can be sick or have some sort of an microorganism growing inside of them and not even know it. So in these cases, the host doesn't go get medical attention because they don't even know they're sick. So these are sometimes called asymptomatic infections or subclinical infections, or just inapparent. Okay, so the microorganism has become established. It has managed to evade the host's defenses, created an infection, multiplied, and now the whole point of the something existing is to multiply and create some sort of offspring, right? So it's got to get out of the host so that it can spread itself to other hosts. So we have these things called portals of exit, just like we had portals of entry. So these are the specific ways that the microorganism, or the pathogen, will say, leaves the host. So examples: secretion, which would be out of some sort of a gland, I suppose. Excretion would be getting rid of wastes, for example, in the urine. That's an excretion. Feces, sort of the same thing. Discharge, which would be like a pus oozing out of a infected wound. Sloughed off tissues like cells, and so on. So the higher the numbers of microbes, the higher the likelihood is that they will create an infection in a new host. So the portals of entry are usually the same as the portals of exit, but sometimes they are different. So a pathogen will enter in one way and exit in a different way. All right, so to recap that whole thing, here's what has to happen. So a microbe has to find a portal of entry, and that's a list of them: skin, GI tract, respiratory tract, etc. It's got to get into the host somehow. It needs to be able to adhere, and it can use fimbriae, capsules, and various things like that to adhere. Then it has to evade and survive the host defenses, so it can't be phagocytized or lysed by other things like complement and things like that. Then it has to cause some sort of a disease or damage, so toxins, enzymes, or indirect damage due to the excessive response of the host's immune system. After all that's been done, it then needs to propagate itself, and so it's got to exit the host and be able to be spread to another organism. Just because an infection appears to be gone doesn't always mean that it is gone. So a person can be quote unquote recovered from some sort of a sickness, meaning they don't have symptoms anymore, but they can still have the microorganism present in them, and there's varying degrees of that. So this term called latency, which is used in a lot of different situations, is the dormant state of the microbe in certain chronic infectious diseases. So an example of several examples, I guess, of viral latency is herpes simplex and herpes zoster. So those are the herpes viruses that cause cold sores, shingles, chicken pox, all that sort of stuff. Shingles is the chicken pox virus later on, when it reemerges later on in life. Hepatitis B, AIDS, Epstein-Barr, all of these viruses, with the possible exception of AIDS, I've just recently read, um, all of these will live in the host for the rest of the host's life. Although they may never potentially ever cause an infection again, they never completely go away. But most of these will reemerge to some degree. The one that's probably the least likely to reemerge would be shingles. So bacterial or protozoan latency, examples of that would be syphilis, typhoid fever, TB, malaria, which is a protozoan. So again, these things can live in you and reemerge later on. This term sequelae, or some people pronounce it sequela. This is essentially the long-term or permanent damage that's caused by some sort of an infection. For example, meningitis can lead to deafness, which never goes away. Strep throat can lead to rheumatic heart disease because of the streptococcal bacteria migrating to the heart valves and colonizing there, which would then result in valve damage. Lyme disease can cause arthritis. Polio can lead to paralysis and just musculoskeletal disfigurement, sort of. So this sequela, S-E-Q-U-I, means follow. So it's the condition that follows an infection. So it's the chronic condition that follows the acute condition. So what happens in your body? So there's 4 phases of infection and disease. The first one is the incubation period. The second period is called the prodrome. The third is called the period of invasion. And the fourth is called the convalescent period. So let's go through each one of those, starting with the incubation period. So this is the time from the initial contact of the microbe to the time you first begin seeing the symptoms from it. So we're constantly fighting off infection. I probably have mentioned this, but oftentimes you just kind of just don't feel good one day, but then the next day you're fine. Well, you were probably fighting some sort of an infection off, and it never really came to the point of full-blown symptoms. But because your body took care of it. And oftentimes you don't even notice, well, I shouldn't say oftentimes, most of the time, you don't even notice anything at all. So we're constantly fighting things off. If it does end up causing symptoms, then that means it made it through the incubation period. So whatever portal of entry this infectious agent comes in through, once it's in there, it begins to multiply. So there aren't enough of them, and the toxic effects of it aren't to the point where it's actually causing symptoms yet. And this incubation period varies greatly based on a couple of things. One is the host resistance, which is just how well you are able to fight off the infection. Certain things are fairly easy for our body to kill, other things are very difficult or impossible. Another one is the degree of virulence. So we've discussed the virulence factors of various microbes, and the more of those they have, the shorter the incubation period is generally going to be. And then the third one is the distance between the target organ and the portal of entry. So the farther away it is, the more likely the body is going to be able to attack it and kill it before it ever gets there. So the incubation period can range from several hours to several years. Majority of infections somewhere between 2 and 30 days. So from the incubation period, we move on then to the prodromal stage, which is the 1 to 2 day period when the symptoms first begin to appear. So this is kind of signified by this vague feeling of discomfort, head and muscle aches, fatigue, maybe upset stomach in some cases, or probably the best term to describe it is this term called malaise, which just means you feel like crap. Like I just don't feel good. I don't really know why. That's just that feeling of malaise, they call it. And really that malaise feeling you're going to feel with probably every single infection that you're going to get. Well, if you get into the full-blown infection, then they call this the period of invasion. And this is where the microbe is multiplying at very high levels, and therefore it's exhibiting the greatest toxicity, and it is, well established, which means you're fully infected at this point. This is when your body is really showing the effects of it. So fever and other prominent and specific signs and symptoms. And when we say specific signs and symptoms, what we mean is specific to that particular infection. And it's those specific signs and symptoms that are often used to actually diagnose the infection, because they will be unique to maybe just a handful of different things. For example, you wouldn't have diarrhea with the common cold, right? But some of these would be things like cough, rash, diarrhea, loss of muscle control, swelling, jaundice, discharge of certain fluids, severe pain. So all of those would be examples with different types of infections, and you would potentially have several with one infection. You aren't going to have all of them, but like obviously liver damage is going to create a jaundice, or not even necessarily liver damage, but just destruction of red blood cells, and the liver can't necessarily keep up with it, or the spleen, or the liver and the spleen. So extremely variable in length based on whatever type of infection that you have. And then the last one is the convalescent period, and this is recovery. So the immune system begins to respond to the infection and fight it off, and then you begin to actually recover. And during this, of course, your symptoms are going to decline, your strength and health gradually return. So for example, in the common cold, this is usually going to be somewhere between the 7th and 10th day, right? One problem with this convalescent period is the infection isn't gone. It's just being controlled to the point where the symptoms are reducing, but it is still alive and well in great numbers. And because of the symptoms reducing, a lot of people stop taking their antibiotics before the full dose has been taken. And this is a problem because we've mentioned this, but just going back to a previous chapter, um, if you stop taking it before it's been completely destroyed, what happens is the resistant bacteria, or whatever the microbe is, generally it's going to be a bacterium, is able to multiply, and then that particular antibiotic is no longer effective. And not only is that a problem in the person who is infected, but the other problem is you may then pass that on to another person. And so if they get infected with a resistant strain and they go to their doctor and the doctor gives them the standard antibiotic, it's not going to be effective. And so they're going to waste all that time and money, and potentially then infect more people, and it just becomes a major problem. And that's why you're supposed to continue taking the antibiotic all the way through the entire prescriptive dose. So this term transmissibility, this is just the ability to be transmitted, right, or passed on to another person or host. In other words, it's whether or not this thing is contagious. And transmissibility varies with each microbe, and also varies with different phases of different microbes. So the measles, for example, are transmissible during the incubation period. Shigella is transmissible during the invasive period, and hepatitis B is transmissible in all periods. A reservoir. A reservoir is just kind of where something is, right? So it's the primary habitat in the natural world where this pathogen originates. So a lot of times the reservoir is a human or an animal carrier, like I don't know, swine flu. You know, the pig would be the animal reservoir. But it can also be soil, can be water, which could be an actual reservoir, uh, oddly enough, or plants. Okay, plants not very common. So the transmitter then is the individual or object from which the infection is acquired. So wherever you get the infection from. In syphilis, the reservoir and transmitter are the same, which would be a person, right? In hepatitis A, the reservoir is a human and the transmitter is food. They call this fecal oral because a human who's growing hepatitis A um, goes to the bathroom and wipes their rear end, doesn't wash their hands well or at all, and then goes and sticks, meat in your taco. Um, reason I say that is because that happened about 10 years ago in the Salt Lake area. I won't say the name of the restaurant, but anyway, then you get this hepatitis outbreak because then you eat it, and so it's the oral transmission of it. So table 11.6 just lists some reservoirs and transmitters of some different categories, I guess. So let's zoom in on that. So over here on the left side, we have reservoirs, and then this column over here is the transmission examples. So this particular group is living reservoirs. So there's a couple of possible choices. Animals, other than humans, because humans are animals, right? But mammals, birds, reptiles, things like that. So mad cow disease in cattle, swine flu, bird flu, those sorts of things. In humans, that would be another example of an animal or a living reservoir. And you can have either an actively sick human who could cause an infection, or just a carrier, somebody who's not necessarily, and we'll actually talk about that a little bit later, but someone who's not currently sick but can still cause an infection in somebody else. And another example of a living reservoir would be arthropods, so insects, spiders, things like that. So we call these things biological vectors. This is showing a picture of a mosquito, which of course can transmit malaria, right? So the malaria lives inside the mosquito. They come and suck your blood, and the saliva from the mosquito causes an infection in the host. So then down here we have non-living reservoirs, and soil, water, and air are the examples of that. The book talks about TB living in soil, but other things would be like tetanus, right? Which is Clostridium tetani or C. tetani. So let's talk about some of these. So living reservoirs, oftentimes, as we've said, other humans, right? So as we mentioned earlier, a carrier is somebody who has the infection in, or the pathogen in them, but is not necessarily infected. So they may not even know they have it. And that brings us to some of the different types of carriers. So this table right here, 11.7, just is trying to illustrate some of this for you. So let's zoom in on that. So the first one type of carrier would be an asymptomatic carrier. So someone who's infected but doesn't show necessarily any signs of the disease. So example, gonorrhea, genital herpes. These are STDs, sexually transmitted disease. So apparently if you go to the club and you're a dancer, you might have STDs. Of course, I'm joking, but anyway, don't go to the club and, I guess, have unprotected relations, apparently. Anyway, incubating carriers. Again, not necessarily full-blown symptoms. So mono is the example they're showing here. So a person has mono, um, you know, multiplying inside of him, but he doesn't necessarily feel sick. And then he goes and kisses his girlfriend, and then she gets mono. Uh, Carriers that can transmit during the convalescent period would be called convalescent carriers. So they're healing and they're feeling better, but they're still contagious. Now, a lot of times you hear people say, well, I'm not contagious, even though they're not necessarily all the way better. And that's because certain types of things aren't contagious during the convalescent period, but some are. So an example, like we said earlier, hepatitis A is contagious in all periods. So that's the example that they give there. Chronic carriers, that would be people who, again, aren't necessarily sick, but once you get a disease, you're going to have it for the rest of your life. So typhoid fever, tuberculosis. So this lady isn't necessarily feeling any kind of symptoms or anything, or maybe she is, but they're sort of, in remission, so to speak, or something like that. And then she, handles your food and gets you sick. And then the last type of a carrier, this is kind of a stretch whether or not this is a carrier to me, but these are people who that aren't infected. They don't have the infection in them. They're not infected, nor have they ever been infected. Um, but what they're doing is getting the infection on them somehow, on their hands. That's the example of this lady here. So for whatever stupid reason, she's handling this poor lady's bedpan with no gloves. And then she walks over to this guy and touches his shoulder and his blanket, and then he gets infected. So she never gets the infection. She just takes it from here and passes it on to this guy. Um, Pretty strange. And the sad thing is, this happens all the time in healthcare. I was having a rod put in my legs. I broke my femur last summer, and I had all the surgical site plus some serious road rash because it happened on my mountain bike. And one of these nursing assistants came in to change the dressing, and he puts his gloves on, lays out the surgical or the dressing on a little card or something. Then he grabs my tray that I had been eating on in the hospital room, just grabs a hold of it, moves it out of the way, then reaches to the garbage can to move it closer to him, reaches inside the garbage can and brings it over to him, and then proceeds to go and start unwrapping my dressing. And I looked at him and I said, "Are you going to change those gloves?" And he kind of looked at me like, "Why?" And I'm thinking to myself, "You got to be kidding me." Um, so anyway, I hope none of you are doing that because I'm pretty sure most of you or all of you are nursing students. So please be aware. This class should hopefully open your eyes to how um, microbes are transmitted. So they're on everything and everywhere. So if you need to be sterile, you can't be touching things that aren't sterile and then expecting that your gloves are still sterile. You've got to change the gloves. And, get good at using your feet. That's how I open doors, sometimes in bathrooms or whatever, how people do. They'll uh, push it open with their foot or their shoulder, or they'll pull it open with a paper towel and then use their foot to prop it open to get through the door. You don't want to be a total germ freak, like Howie Mandel type, but you need, especially in the healthcare field, you absolutely need to be completely a germaphobe when it comes to that, or you will literally kill people. So, animals as reservoirs. So a biological vector then is a vector that's living, right? And this is one that actively participates in the life cycle of the pathogen. So there's often times where um, a living animal has to be present in order for the pathogen to actually get through its entire life cycle. Sometimes it only needs one, like human, but oftentimes it's got to live in like I've mentioned before, a pig, or another animal like rabies could be anything, a bat, a dog, a raccoon, whatever, but then they can get passed on to a human. A mechanical vector is where it carries the vector like on it, not necessarily in it. A person at my church used to work at the University of Utah, and he was working in a lab that was studying leprosy, and they were growing this stuff on the feet of mice. He said that that's the best. There were other ways they could grow it, but he said the best place and the way it would grow the best in order for them to culture this was on the feet of mice. So that's pretty interesting. So I think that would be probably a type of mechanical vector. Not entirely sure. It might not be, but I think it is. So the majority of animal reservoirs are arthropods. So things like fleas, mosquitoes, so insects, and I think spiders is also part of that as well. But large animals can also spread infection. So as I said, mammals carry rabies. Um, birds carry psittacosis. Lizards can carry salmonellosis. So zoonosis or azoonosis is an infection that's indigenous or natural to animals, but can be transmitted to humans. So the human doesn't actually participate in the life cycle because once it gets to the human, it can't persist after that. But it can infect the human. So they call this being like a dead end host. So your book says this makes up 70% of all of the new emerging diseases worldwide. So the bad thing about this is you can't get rid of the infectious agent without eradicating the animal reservoir. And that also comes with some significant problems because when you eliminate, even if you could, which is probably impossible, when you eliminate a type of animal or insect, it can completely mess up the entire ecosystem. So even though it's something that seems bad, it could create some devastating effects that we wouldn't want in other situations. So worldwide, attempts have been made to get rid of all mosquitoes and certain rodents because they are such prominent carriers of diseases. So there are some common zoonotic or zoonotic infections that are listed here in table 11.8. We don't need to go through all these. A lot of these you already know. Mammals carry rabies, so showing a picture of raccoons here. The hantavirus, that was a big deal about 15 years ago. I remember hearing on the news because mice and rats carry it. And so people, I don't know if they were actually getting it to a higher degree, but I remember down south, like Arizona area, they were talking about it. Influenza, certain types of flu, and there are thousands or millions of types of influenza, I don't know, but chickens, the avian flu, swine flu on pigs. Mosquitoes carry the West Nile virus. That was also a big thing. I think that these things just get, in news cycles sometimes and aren't necessarily more prominent or prevalent, but for some reason I think the news needs something to talk about, and so they start kind of uh, bringing this stuff to the forefront, and then all of a sudden all the other news sources jump on the bandwagon, and before you know it, everybody's paranoid of something that hasn't really even changed. Anyway, that list goes on, and you can read through that. This one, maybe I should mention toxoplasmosis cats. This is the one that pregnant women are warned against because they say that pregnant women aren't supposed to be changing litter boxes because it creates, I guess it can infect the fetus in a pregnant woman, and it's passed on by cat fecal matter. All right, so non-living reservoirs. Most of the microbes that live in soil and water are saprobes. which you remember live on dead, decaying matter, right? And these things generally don't cause any harm to humans, and to the contrary, uh, cause great benefit to humans. They break down this organic matter so then it can be used for things like fertilizer, and just getting rid of dead stuff. Otherwise, it would just keep piling up, right? Some are opportunists, which means they don't need humans, but when they have the ability to colonize, they'll take it. And a few of them that live in these non-living reservoirs are actual, regular pathogens. Protozoa are a very good example of that, live in, pond water and things like that. So, how do we acquire and transmit these infectious agents? Let's talk about a few terms and define them. The first one is called communicable. So, something that is communicable is something that can be passed on and established in another host. Now, infectious oftentimes is used synonymously with communicable, but technically it is not. And I'm not entirely clear on the difference anyway, so you're going to hear me use it interchangeably. If it's contagious, that means that it is highly communicable. In other words, the likelihood of you getting an infection is very, very great. There's lots and lots of things that are communicable but aren't necessarily contagious. When we're introduced to communicable diseases, probably, I don't know, I'm just guessing, probably every single day, but certainly a lot. But they're not necessarily contagious. So influenza or the flu and measles are examples of things that are highly contagious. Hansen's disease, which I guess is leprosy, is only weakly communicable. So non-communicable diseases is a disease that is infectious, but does not arise through the transmission from one person to another. So going back to what we said earlier, infectious and communicable not being the same thing, because you can have a non-communicable infection. So I hope that makes sense. But again, watch for me to screw that up because I promise I will. But let's talk about what that is, because right now I'm sure this is not making any sense. The primary way is when a compromised person gets invaded by their own microbiota, which we called opportunistic infections from a previous chapter, or an individual having contact with a microbe that exists in a non-living reservoir. And then it begins to multiply and colonize and create an infection. So important to know, with a non-communicable disease, a person who is sick with it is not going to cause an infection in another person. If you think about it, the other person probably already has that microbe living on or in them anyway, and they're not sick. That's because they don't have a compromised immune system, right? So anyway, infectious can be communicable or non-communicable. So patterns of transmission in communicable diseases. They can be direct or indirect contact with either animate, which is living, or inanimate objects. So the first type is called horizontal transfer. And this is where it's transmitted from one person to another, or from, one similar animal to another similar animal, in veterinary or whatever examples. But anyway, it goes from one like thing to the same thing or similar thing, right? Vertical transfer is from parent to offspring. And there's two major modes of transmission, either direct contact or indirect transmission, where there's some sort of a vector or a vehicle involved, like the bedpan that lady showed. So the first lady in the bed and that would be an example, of course, of horizontal transfer. Um, she would be the infected individual, and then the second person was the guy in the other bed who, the vehicle was the bedpan that the lady brought over, or her hands, I guess. It could be like a cup or something. If you drink after somebody, that could be the vehicle. Anyway, biting vectors are these arthropods, generally, that can transmit the disease through the injection of their saliva, like in the case of a mosquito, or fleas, which apparently defecate around the bite wound, or in the case of the tsetse fly, they regurgitate blood into the wound. Either way, those things would cause an infection. Mechanical vectors, not necessary to the life cycle. That would be like the bedpan. Certainly, you don't need a bedpan to cause, I don't know, hepatitis or something. But it can be used to carry the infection. So external body parts are an example. Food, we had mentioned earlier, would be another example. House flies is a good example of a mechanical vector because they fly around and then go and land on, dog poo or whatever, and get the E. coli bacteria or whatever on them, on their feet, on their mouth parts, and then they go and fly on your food and start doing the same thing to your fried chicken at your picnic, and the next thing you know, you're eating, what was on the dog poo. So it's pretty disgusting. And houseflies in particular are attracted to foul-smelling things, which are generally rotten or dying, rotten, or fecal matter. So that's why they're particularly in good at causing infection. Okay, so this table here just shows some patterns of these communicable diseases. So the first one here is direct transmission, and this is from contact from one person to another. This is showing, sorry, that's in the way there, but Epstein-Barr virus, which is what causes mono. One of the things it causes is mononucleosis, which they call the kissing disease, right? But there are other forms of direct transmission, such as sexual intercourse, right? So touching, kissing, sex, which that should say intercourse because sex technically means gender in anatomy. But anyway, another example is what this lady's doing over here. She's apparently spewing a mouthful of water, which she's really not. She's just coughing or sneezing, but it looks like she's spitting water all over that lady. Anyway, that's obviously another way of transmitting infection, like the common cold or flus and things like that. And I missed this, sorry. So that was horizontal transmission, and we'll come back to that in a second, but I missed the vertical. Remember, vertical is from parent to offspring. So either into the egg via the sperm, or passing from the mom through the placenta into the fetus, or passing through the milk in the infant. Okay, but back to horizontal. So we had two types. We had direct, and then we have indirect transmission. So this is where it goes from one person to another, but there has to be something in between, which they're calling the intermediate conveyor. Okay, so there's two types. There's what's called a fomite, which is an inanimate object, not a living animal or any kind of a living organism. So it's something that the pathogen can live on, at least for a short period. Huge one here, doorknobs. That's why, you know, more and more commonly you're seeing these days garbage cans by the door of bathrooms because people are becoming more and more aware of this. And I mean, it's disgusting. If you go into a men's bathroom, they say like over 70% of men don't wash their hands after using the bathroom. Um, I'm sure that's increased when there's another person in the bathroom. It's astounding to me how many men will just walk out of the bathroom even after using, the stall, which women may not understand this, but the stall generally implies that they went number two, and they'll just walk right out. Oftentimes they do the fake hand washing, where they walk over and stick their hand under the water for about 2 seconds and fling off the water. I mean, it's absolutely ridiculous and disgusting, and it makes you think Howie Mandel might have something with not shaking anyone's hands. In a way, I don't blame him, but I just try to wash my hands a lot. Um, anyway, that's why I don't touch doorknobs very often. But anyway, it can be anything like that. Faucet handles is also common. I always tell my kids when they're washing their hands, to turn the water off with their elbow or with the paper towel or something like that, because you wash your hands and then you turn right back around and touch the faucet that, when you turned it on, your hands and whoever else's hands were dirty. Anyway, that's all the kind of stuff you guys need to really, really be thinking about as healthcare providers. So that's a fomite. A vehicle. So fomites, by the way, are not natural things. They're not things that exist in nature. Doorknobs, telephone receivers, faucet handles, those don't exist in nature. Vehicles are also non-living, but they do exist in nature, such as air, water, soil, food, and you can read through that and see all the things that go along with that. Now, your book lists something called a special category called oral fecal, which I learned this as fecal oral because you don't infect feces. You don't go from the mouth to the feces, you go from the feces to the mouth, and that's how you get an infection. So I'm not sure why they call it oral fecal, but anyway, obviously this is where, like hepatitis is the best example, where um, the food handler has feces on, his or her hands and then puts it in your taco or hamburger or whatever, and then you get sick. So it goes from the feces to your mouth, and then that's how you get an infection. So I'm not sure why this is a special category, but I guess because it's semi-natural. I mean, it's natural, but it's not necessarily a vehicle like, air, water, or soil because it's created from a person. I don't know. It probably fits a little better to me up in the direct contact, but either way, it's certainly horizontal, so it kind of doesn't fit very well in direct or indirect. It's kind of in the middle somewhere. So anyway, fecal oral transmission or oral fecal. Okay, so another really important thing to talk about is something called nosocomial infections. And nosocomial infections are um, infections. It says hospital, but that not necessarily a hospital, just any kind of a health care, sort of related infection. Okay, so they're also called healthcare associated or iatrogenic infections, and these are developed while you are being provided for by some sort of a healthcare provider. Okay, typically, yes, it's in a hospital, but it could be at a dentist's office, it could be even at a home health care, whatever. If the person providing your healthcare does something they shouldn't, or even not necessarily anything they shouldn't, it's just sort of the risk involved. Um, and if you get an infection from that, then it's called a nosocomial infection or an iatrogenic infection. So somewhere between 0.1% and 20% of all admitted patients get a nosocomial infection. Average is about 5%. Um, and that equates to about 2 to 4 million cases per year, and that results in 90,000 deaths. So if you are a nursing student or a nurse, I'm talking about you, okay? Um, it's the doctors and the nurses who are causing these infections oftentimes. Now, like I said, it's not always. Sometimes it is just bad luck. I mean, They have filtered air systems sometimes, but sometimes they don't. And if the furnace is spewing out, you know, some sort of an infection all throughout the hospital or the nursing home or whatever the case may be, that's not necessarily someone's fault, but oftentimes it is. The example I gave of the nurse's assistant um, not changing his gloves. Um, that's certainly something that is controllable. Anyway, it equates to 8 million additional days of hospitalization and costs somewhere between 5 and 10 billion dollars annually. So this is a major, major, major problem. Some sources that I have read put this as the well, that's not true, um, including improper prescription of medications like drug interactions, or the wrong drug, or those sorts of things. Combining those with this, some sources I have read say that it's the #1 cause of death in America. So what are some of the things that influence these infections? One, of course, is the fact that oftentimes these people who are in the hospital are already compromised, right? If you've got, for example, an AIDS patient in the hospital, the chances of them getting a nosocomial infection are drastically higher than somebody who's in there, because they broke their leg or something like that. Another reason is because it's a collection point for pathogens. In other words, there's a lot of sick people in the hospital. So the odds of somebody getting sick are much greater than if you are staying in, a cabin up, in the Rocky Mountains all by yourself. There's no one even around you. So what are the odds of you getting some sort of an infection from somebody? Pretty much zero, right? So of course the chances are greater if you're in a hospital. Um, I guess the point they're trying to make here is opportunistic infections of your normal biota because of a decreased resistance to being able to fight them off. And then fomite infection is also obviously going to be higher because you're getting poked and prodded with a whole bunch of stuff, especially stuff that potentially has pathogens on it. So there are certain processes that are more likely to cause a nosocomial infection. For example, treatment using reusable instruments such as respirators, thermometers, and those sorts of things. Now, nowadays, we're pretty darn good in our healthcare system at making sure all of those sorts of things are sterile. But a lot of times, accidents occur. A person doesn't realize something has been used, for example, and they use it on a second person. Also, things that aren't necessarily anything we can do much about. Catheters. Catheters are just, they just make it easier for microbes to grow on, and therefore it's just an inherent risk with a catheter that they could get, a urinary tract infection. Heart valves, prosthetic heart valves. So if you have a valve replacement surgery, grafts like skin grafts or bone grafts and those sorts of things, it's very likely that a pathogen can be in the graft, and it's impossible to sterilize a living tissue, right? Drainage tubes, trachs, those sorts of things. All of those things are going to make it more likely for a pathogen to be able to enter, and that's why you have to change them regularly. And they don't even like to keep them in for very long, and that's partially the reason why. Another thing is that a very high proportion of people in the hospital are getting, antibiotic therapy. So there's a lot greater chance that there are going to be antibiotic resistant uh, microbes in the hospital. And so it's just a concentration of these more resistant bacteria or other microbes. So and again, the likelihood of getting a nosocomial infection is greater. So the most common nosocomial infections are UTIs, respiratory infections, and infection at the surgical incision. MRSA is an example of that methicillin-resistant Staphylococcus aureus, right? Serious infection. Matter of fact, my wife's grandfather just last week had his foot and lower leg removed, had amputated because of a MRSA infection. So scary, bad stuff. And his followed a foot surgery. He had some bones fused because of neuropathy in his foot, and he ended up with an iatrogenic infection and lost his foot. Another common one is gram-negative intestinal biota like E. coli. So they've found these in more than half of the patients with nosocomial infections. Another one are gram-positive bacteria like staph, that would be MRSA, strep, and then of course not gram-positive bacteria, but another one is yeasts. So something to make note of, staph, and to a large degree strep, as well as E. coli, these are all normal biota. These live on pretty much everybody, right? The point is, if they, if E. coli lives in your colon, it's fine. But if E. coli is introduced into your mouth, we got a problem, right? But there are true pathogens that can also be transmitted, such as tuberculosis, salmonella, Hep B, the flu, and those things are not normal flora or biota, right? So this right here just shows a pie chart of the percentages of these nosocomial infections. So you can see UTIs make up the vast majority, and that is because primarily of the catheters, right? The next biggest one would be infection at the surgical site, and I personally think that the vast majority of these are preventable. I think that people just don't realize, even though they've supposedly had education, I don't think they realize how these pathogens are actually passed on. So I would hope that you guys are thinking about this often so that you will prevent this. Respiratory, again, there's a couple of reasons why respiratory is so high. One is because the person is sedentary, and that leads to pneumonia. Hip fracture is one of the highest causes of death in elderly. And the funny thing is, it's not the actual fracture that's the problem. It's the stuff that results from the fracture. For example, surgical complications, infection from the wound, stroke caused by blood clots forming while the person is actually healing. And then another huge part of that is respiratory infections, because when you're sedentary, it causes pneumonia. So a huge percentage of elderly who have hip fractures end up dying of pneumonia because they can't get out of bed. Anyway, other meningitis, gastroenteritis, skin infections, septicemia, which is a blood infection. It kind of rounds the thing out. So how do we control these? Well, I did not realize this, but apparently Medicaid and Medicare don't reimburse hospitals for nosocomial catheter-related UTIs because apparently they feel like they are preventable. They also don't pay for vascular catheter-associated infections, which would be IVs, or surgical site infections, because they consider these all to be preventable. So apparently it's the hospital that ends up paying for these things. So it would behoove the healthcare providers to be extra careful because these costs related with these things, as we said earlier, are in the billions every year, right? Um, so a lot of hospitals apparently have what is called an infection control officer, which is somebody who just keeps track of the policies and procedures and the statistics relating to these nosocomial infections. And they're also in charge of training the healthcare providers in aseptic technique, which of course you guys know is the techniques involved in keeping infections from occurring. So the people who are in direct contact with these people and have a higher chance of um, passing these on because of needle sticks or being in contact with infectious secretions, bloods, and those sorts of things, they need to be especially aware of how to control these infections. You know, the secretary at the front desk, not so much. But if you're taking this class, you're not a secretary at the front desk, most likely. You are probably a nurse. Okay, so we have these universal blood and body fluid precautions that have been come up with. So more than a third of nosocomial infections can be avoided by consistent and rigorous infection control methods that we are going to talk about right now. So there's 6 of them. The first one are barrier precautions. So that's just creating a barrier. So masks and gloves should be used to prevent the contact of skin and mucous membranes with um, an infected patient, right? So um, if you're going to be touching somebody's wound, you want to put gloves on, obviously. And I'm shocked. I was just talking to a patient of mine who is a nurse, and she was telling me how she has seen people change dressings without gloves on. She says it's mainly the older people, older nurses, but that is absolutely unacceptable. That person should be fired, in my opinion. Anyway, double gloving is also a good idea. I don't necessarily think you need a double glove all the time, but apparently what happens is the gloves actually can get microscopic holes in them, maybe from manufacturing, or maybe just, you touch something and it pokes a hole in it and you don't even realize it's in there. Um, so that's something you need to be aware of, but that's also why you wash your hands after you take the gloves off too. Other barriers would be gowns, aprons, other body coverings. You've probably seen those face shields that they put on. And these should all be worn during surgeries, venipuncture. You should obviously put gloves on when you're giving somebody an IV, or of course, emergency procedures. Dental workers should wear eyewear because they're flicking stuff up in the air sometimes. Even just flossing someone's teeth could flip something in your eye, for example, or in your mouth. That's why the dental workers should always have on a mask and some sort of eye protection. Or in some cases, face shields. And that's, like I said, to keep that stuff out of your face. The second one is sharps precautions. So this is just being careful with sharp things, needles, scalpels, other sharp devices. So when they're used on a person, then obviously they're contaminated, and you've got to be extremely careful to dispose of them very, very quickly and safely so that they're not going to infect you or another patient. So you should never try to put a cap back on a syringe. That used to be common practice a long time ago. Nowadays, they have needles that, which you guys all know this better than I do, actually. Um, but they have these needles that have a little plastic thing that pops up over the needle after it's been used, and that's just obviously to prevent it from poking somebody else. One of the common ones was actually invented by a company up in northern Utah. And then, of course, you shouldn't just leave, unprotected needles just laying around. So reusable needles, which I didn't even know we had those anymore, or other sharp devices, must be heat sterilized in some sort of a puncture-proof holder before they're handled. So they're sterilized. I think what they do is they put them in a, they probably double sterilize them. They sterilize them, then they put them in a package and seal them. Then they re-sterilize them. And that way they know if they're in a package, they're not going to be contaminated until the package is opened again. The third one are dental precautions. So of course, dental instruments should be sterilized between patients. And I mean, this didn't used to happen. Seems crazy now, but before they knew about this stuff, they just went from one patient to the other. If sterilization is not possible, they should be, of course, thoroughly disinfected with high-level disinfection like um, peroxide or sodium hypochlorite or potentially bleach if that is possible. Certainly bleach is good on countertops and things like that, right? Blood and saliva should be removed completely. So they should be, what we're saying is you should wash them or rinse them before you sterilize them. I mean, you're not going to throw, use some tool in someone's colon and get fecal matter on it and then just throw it in the autoclave without taking the fecal matter off. Yeah, it's going to be sterile, but it's still going to be disgusting, right? So point is you just wash that stuff off. The fourth one, hand washing. So you should use germicidal soap, and we're not going to teach you how to wash your hands in this class. That's something that if you're a nurse you already should know. But using germicidal soap, scrubbing, using brushes, washing for a certain period of time, making sure you get in between your fingers, making sure you're not wearing jewelry on your hands. All of those things are things you need to be aware of when you're washing your hands. So it says here when you've accidentally contaminated, well, obviously, but also when you not necessarily just when you think you've been contaminated, just all the time you should be washing your hands. So you take your gloves off, whatever, before you see a patient, you should be regularly washing your hands because you don't necessarily know when you've been contaminated. Saliva precautions, number 5. So this, goes along with dental, but also, trach tubes and things like that, or feeding tubes. At least the oral kind. Because saliva can be a source of certain infections, you should have some sort of a barrier with, and this is specifically talking about resuscitation. So we generally in the hospital setting, no one's going to do mouth-to-mouth resuscitation. They put a bag over the mouth and use the bag to do the breaths with, right? But out in the field, you may do that. You may have seen those barriers, little plastic barriers that you can put in your wallet or your purse. So if you're a nurse and you know CPR, well, if you're a nurse, you do know CPR because you have to. But a lot of people carry these portable, disposable barriers. So if they ever run into somebody whom they have to do mouth-to-mouth resuscitation on, they can use a barrier. I would say the likelihood of you getting sick is not very common doing mouth-to-mouth resuscitation, but it's possible. And number 6, healthcare worker precautions. So if you have active lesions, you should not be handling any patients or equipment. Okay, and I'm sure that's, what do you consider a lesion? What if you have a zit? Technically that's a lesion, right? I don't know. Certainly if you've got, I don't know what, maybe cold sores, I don't know, maybe not. Probably varies from hospital to hospital. If you've got a cut on your hand, should you be handling patients? Well, I guess maybe it depends on how big the cut is. I don't know. I would say any cut. But gosh, how many times do we have cuts on our hands? I mean, I've probably got cuts on my hands 75% of the time. So I don't know, like where you draw the line here. Pregnant healthcare workers need to be extra diligent so that you don't run the risk of causing an infection of your fetus. And of course, it's recommended that healthcare providers are vaccinated. Not necessarily even not recommended. I mean, it's required. So you'll have a certain list of vaccines that you have to have if you're a healthcare worker, such as Hep, A, B, and C. I think all three. In some cases, anthrax, tetanus, which isn't technically a vaccine, but it's a you have to have the booster shots and so on. Okay, so obviously I didn't do a very good job of transitioning to this next thing we're going to talk about, but we're moving away from procedures to just determining what is causing the some sort of an infection. So we use these things. We had mentioned this, I think, in chapter 1. These things called Koch's postulates. So these things are just what was his name? Robert Koch, I think, came up with these techniques to determine what is causing the infection. And that word we use for that is the etiology. So it's the etiologic or causative agent, which is just the cause of the infection or the disease. So Koch's postulates are a series of proofs that this guy came up with to determine the cause of these diseases, and we still use them today to determine etiology. So they're reliable for many diseases, but not all of them. And the reason why is we don't necessarily have the ability to perform cultures of every single type of disease, and so we aren't able to isolate every type of pathogen. So they don't work for everything, but ones that we can isolate in culture, we can use them for. Which apparently I got ahead of myself. So that's what I was just saying. You can't grow them in the lab. Some of them we can't culture on other animals because they only infect humans, and we of course can't use humans as laboratory test animals. And when there's a polymicrobial disease, there's no way to use Koch's postulates because part of Koch's postulates requires that we isolate a single microbe. Therefore, if it's a polymicrobial disease, that pretty much eliminates the possibility of using Koch's postulates for that. But table 11.1 shows how Koch's postulates works absolutely perfectly. So let's go through that. Okay, so the first thing is you're going to find a particular microbe in every case of some certain disease. Lyme disease, tuberculosis, anthrax, whatever the case may be, you're going to run into some certain thing. Now, you may think to yourself, well, then that's what causes it. But that is not necessarily true, because you may find something in every case of the disease, but it's just coincidence, and it's not actually causing the disease. Or it may be like an opportunistic infection, and still, again, not causing the disease. So the important thing is we have to see if it's causing the disease. So just because it's present doesn't bring us to the conclusion that that's what's causing the disease, but it certainly is the first part, okay? So we take this thing, maybe a body fluid, saliva, for example. Then what we do is we isolate it using the culture techniques that we learned in the previous chapters of this book and the lab associated with this class. So we isolate it in the lab. This is showing a petri dish, but it wouldn't necessarily be a petri dish. It could be test tubes or whatever. And once we isolate it, we of course then we classify it and determine what that thing is. So we use all of the techniques we've learned in previous chapters to identify the microbe based on things like morphology and genetics um, gram testing, all of those sorts of things. So once we have isolated this thing, we then proceed to determine if this is the thing that actually causes the disease. So what we do then is we find a healthy subject, which if, again, if the only thing that it infects is a human, it's impossible for us to do this, right? But if it will infect a lab animal like a mouse, we go ahead and re-inoculate it, which is what's showing the picture of there. And then we see if the thing we isolated in this previous slide here, right here, we see if it causes the disease in the lab animal. And if it does, we then try to isolate again and see if we find that same thing. And if we are able to cultivate out of this thing and culture it and determine that this thing here in step 4 is the same as the thing we found in step 2, then we can postulate, of course, that the original microbe that we got from step 1 is the actual thing that is causing the disease. And at that point, once we know the etiology, we can then proceed to find some sort of a therapy uh, for that particular thing, which would be like an antibiotic or something. Okay, for each of the descriptions below, determine if it pertains to an exotoxin or an endotoxin. So this is going clear back to table 11.4. So you may want to look back and forth on that while we're going through this. Okay, so toxic in small amounts would be exotoxin. The one that causes systemic effects like fever and inflammation would be endotoxin. The one released by the cell when it sheds its outer membranes during lysis would be, again, endotoxin. The one that's composed of small proteins would be exotoxin, which means the one that's made of lipopolysaccharide, or LPS, is endotoxin. And the one that can be converted into a toxoid would be exotoxin. Endotoxin cannot. Okay, so a very, very, very important field of study associated with disease is this thing called epidemiology. And epidemiology is just the study of disease in populations. So it's the study of the frequency, the distribution, and other related factors in populations. In other words, what we're seeing is, where it is, how it spreads, how toxic it is, also all of those sorts of things. So it involves many disciplines, including microbiology, anatomy, physiology, immunology, medicine, psychology, sociology, ecology, statistics, um, because all of those things are associated with how a disease spreads. We're not going to talk about all of those, but an epidemiologist would be concerned with all these. For example, why would a certain disease, be more prevalent in a certain socioeconomic, class? Maybe how do I say this? I'll say it like this. Maybe like low income housing. Okay. Why are more people getting, HIV? Well, I don't know. Maybe unprotected sex, maybe, drug usage, heroin, things like that. Um, maybe poor nutrition. mean, the thing is, it's probably multiples of things all put together, right? Not to say everybody in low-income housing is that. I'm just saying that that's certainly just by nature of the beast, just unfortunately more common, you know, in those certain situations. I mean, you may also have certain diseases that are more common in high socioeconomics. I don't know. But anyway, the point is there's a whole bunch of things that need to be considered. It also considers all forms of disease. Example, heart disease, cancer, drug addiction, mental illness. Now, for this class, we're not going to talk about all of those. We're not concerned with cancer or drug addiction, right? We're going to be focusing on diseases caused by pathogens because this is microbiology, okay? So there's this lady you may have heard of. Her name is Florence Nightingale. And this lady sort of laid the groundwork for, what is now the study of epidemiology. So this she figured out even before the germ theory. So this is before they even knew there were such things as pathogens. But she figured out that filth, just lack of sanitation caused diseases. She didn't even know why, okay? But she instituted methods in military field hospitals of sanitation, okay? So using separate linens and towels for each patient, they would clean them and then use clean ones for each patient. Cleaning the floors, unclogging sewage pipes. Seems obvious, but apparently it wasn't. And she also kept really good notes that demonstrated how more people, men, I guess, because that's all that fought in the wars back then, more men died of the disease rather than the actual traumatic injury. So, back in the day, they would just chop your leg off because you'd end up with gangrene, because they didn't even, first of all, know what it was. Second of all, didn't know how it was spreading, which what it was spreading from is because they were using the same surgical instruments on every single person. And so they're just passing it from one person to the next. Anyway, she took note of all of this and sort of discovered it even before she understood what was actually causing it. Okay, but back to this epidemiology. How do we, one of the things we do is we have to track these diseases in a population. So these aren't all tracked. We don't track the common cold, right? Most people don't even go to the doctor when you have, a cold. But some of them we do, and these are called reportable or notifiable diseases. And so these are diseases that must be reported to the authorities, primarily the CDC, which is the Centers for Disease Control. Other diseases are reported on a voluntary basis. And when we say reported, this is by the healthcare provider. This is not by you. So we have these networks built up consisting of local, district, state, national, and international agencies that all work together to track these diseases. And table 11.11 lists the reportable diseases that are reportable to the CDC or just your local health department, which will eventually get to the CDC. Now, I don't expect you to remember this, but it sure wouldn't be a bad idea to run down through this list just so that you're familiar with it. Most of these would be fairly obvious, okay? Anthrax, the flu right here, Haemophilus influenzae, leprosy, which is Hansen's disease, hantavirus, HIV, obvious. Now, some of them maybe not so obvious. Botulism, which is Clostridium botulinum. That's caused from usually foodborne illness. So, foodborne illnesses are important because it could be coming from a source like a grocery store. Just recently, what do they call those? A bulk chain had some berries, frozen berries that were causing an infection. And so thankfully these things are reportable. And once they start seeing several cases, they start trying to track down where they came from. And what they discovered was they were coming from these frozen berries from this store. And so then, of course, they recalled the berries. So that's why those foodborne illnesses are reportable. And then, of course, uh, smallpox and things like that right there, these are supposed to have been eradicated, right? So if we see a case of smallpox, we've got a major problem. So obviously that's going to remain on there forever. But anyway, run down through that and make yourself a little bit familiar with that. And always keep that as a reference as well. I don't know how these are reported as far as in the healthcare setting. It would probably vary based on the hospital to hospital, but I would suppose that nurses are probably responsible for entering these into the database. So something you definitely want to be aware of. Okay, so how about statistics regarding epidemiology? So one of the things that is tracked is obviously the frequency of the cases. And so there's a couple of terms that we're going to be familiar with here. One is called the prevalence. The prevalence is the total number of existing cases with respect to the entire population. In other words, it's just the percentage of the population who has a particular disease at a particular time. And this is calculated fairly simply. You just take the total number of cases in a population and divide it by the number of persons in a population, and that gives you a decimal, and then you multiply it by 100. So for example, let's say you have 100 people in a town, and 50% of them have an infection. 50 divided by 100 is 0.5. You multiply that by 100, and obviously the number you're going to get is 50%, right? So that would be the prevalence. The incidence is a little different, okay? It's also known as the case rate or the morbidity rate. And this measures the number of new cases over a time period. So from this, we can get the rate of infection and also the risk of infection. And that just helps us determine basically how infectious something is. So how do we do that? Or how do we get this number? We take the number of new cases over a time period, so maybe a month or something, and then we divide by that, or divide that by the number of susceptible persons. So obviously excluded from that would be the people who already have the disease, right? Because that they aren't going to get infected again. They're already infected, so we wouldn't include those. But we're going to get a number which is the incidence. Okay, so we usually report this per 100,000. So let me think about this for a second, see if I can make sense of this. So let's say you have the number you get is 0.1. No, let's make that a little easier. Let's say it's 0.01. When you take the number of new cases and divide it by the number of susceptible people in a population, and you get 0.01, which is 1/100th. Basically what you do is you just add zeros to the end of that until you get to the 100,000th mark, and that's going to be your incidence, which you might verify this because I could be wrong, but if my math is working in my brain right now, 0.01 would be 1 per 100, which would be 10 per 1,000, which would be 100 per 10,000, which would be 1,000 per 100,000. So the incidence would be 1,000 per 100,000 people. I hope that made sense and didn't just confuse you more. Anyway, these are usually very small numbers, so it's usually going to be something like 0.2 per 100,000 people. So they'll follow the incidence and the prevalence over a seasonal, yearly, or long-term basis. and they can see from that trends. For example, the common cold is more common, I guess, in the what we call the cold and flu season, which is in the wintertime. Same with the flu. Um, I've heard different theories as to why that is, such as that it cycles around the world and like comes back every season, and I don't buy that at all. What I think is significantly more likely, and this is based on a lot of research, is the fact that during the winter time, three things happen. One is people are staying indoors more, which means we're more confined. So we're able to pass these infections on to each other much more easily. Two is that since people are more infected, we're going to pass it on more easily. Okay, so it's that those sort of go together. But the other reason is because vitamin D is a very significant nutrient regarding your immune system. And in the summertime, and this is obviously in the more northern and southern hemispheres, not near the equator, there's no cold and flu season near the equator. Okay, did you ever ask yourself why? Well, it's not because the supposed uh, flu virus doesn't go around the world. It somehow misses the equator, at least in my opinion and a lot of other people's opinions. It's because in the northern and southern hemispheres, the sun in this in the summertime of the northern hemisphere is down in the southern hemisphere, and what happens is it's further away, and it is also at an angle that causes most of the UVB rays to actually bounce off of the atmosphere and never even make it to us. So a little bit gets through, certainly some of the UVA, because we definitely can get sunburned in the summer, but not nearly to the degree. So that's part of the problem. We don't get the UVB, and of course you may know the UVB rays are responsible for causing the vitamin D synthesis from the cholesterol in our skin, right? But the other reason is because it's too cold, and so A, we're not spending as much time outdoors, and B, when we are outdoors, we're not out there in t-shirts and shorts. So we're covered up, so there's not any skin really even showing. So all that basically equates to less vitamin D production. So anyway, I think that's more likely the reason why we get this supposed cold and flu season. So it's very important to take a high quality vitamin D3 cholecalciferol supplement. Anyway, a little off the subject. So let's get back to epidemiology. Okay, so statistics of concern are the rates of the disease with regard to gender, race, or geographical region, because they can figure out these patterns and see why a certain person's getting sick. Race, like example of that would be sickle cell anemia, almost primarily involved in African Americans, right? So one of the things that's important in that, I work with a college team as one of the physicians, and all of the African American athletes have to be tested for sickle cell anemia to find out whether they're a carrier or actually have sickle cell anemia. And the only reason they know this, obviously, is from epidemiology. Okay. Well, there's some other things, some other terms that we have to define here. One of them is called mortality rate, and this measures the total number of deaths due to some specific disease. Another one is called the morbidity rate, and this is the number that represents the people that have the infectious disease. So the morbidity is the number of people infected. The mortality is the number of people who die. So the overall death rate or the mortality rate worldwide has dropped of just all the infectious diseases, but the morbidity rate hasn't. what that tells us is the same number of people are getting infected. So we're not doing a very good job of reducing infections, but we're doing a pretty decent job at curing them. Okay. So this right here is an example of a graph of the statistics of hepatitis. Okay, so this is the incidence right here of hepatitis, and this is in the United States for this 40-year period right here. So all infectious agents are going to have some sort of a graph like this, and probably a lot of them are going to follow a fairly similar trend. But this is all three forms of hepatitis. So the green line is A, the blue line is B, and then the red line is C. And you can see that all of them have dramatically decreased, and that's because of awareness, sanitation, and those sorts of things. So like I said, you're going to see a similar graph for every single infectious disease pretty much. And one interesting thing is that apparently we weren't tracking hepatitis C prior to 1990. So another example of a graph, epidemiological graph, is HPV, which is human papillomavirus. So the virus that causes warts, which is including genital warts. And this is showing the prevalence, right, which is the percentage of the population. So this is broken up demographically, 20 to 24 age and 25 to 29, which is what, both of these are 4 years, right? Well, I guess 5 if you include, yeah, so 5 years each. And you can see that the younger group is significantly higher than the older group, and probably just because of, I mean, my assumption would be just because of general maturity. But this just shows how this can be broken up and how it's so significantly different. White lower than black, and black higher than Mexican. Not exactly sure why that would be, but certainly there's probably a reason. Education, not a significant difference. High school diploma, not having a high school diploma, and some college or more. You can see that the people who do have some sort of college education is slightly lower. Again, if this is tracking genital warts, which I'm assuming it is, that's a sexually transmitted disease. So probably some education would be, equated with, I don't know, higher intelligence isn't the right word, but probably certainly less ignorance. Apparently, the lower the income, the higher the incidence. And then this one is the big one, the number of sexual partners. If you have 3 or more sexual partners, you're significantly more likely to have genital warts than if you have 1. So, moral of the story, don't be sleeping around. Here's another type of a graph. I don't know if you call it a graph. This is a chart, I suppose. Is showing the incidence of malaria. So everywhere where you see green is no known malaria. So if you look in the United States, apparently there are 0 cases of malaria. Notice something. All of these cases where malaria is significantly present is all around the equator. Now, this says malaria presence varies in these states here, or these countries here, probably because of just the reportability is, or the reporting is just spotty. But anyway, you can see that it's primarily around the equator there. Okay, so there are these different trends that we see in these statistics, and what these tell us are the way that these certain diseases are spread, and that helps epidemiologists know where to look when they're trying to control something. So the first one is called a point source epidemic. And in a point source epidemic, the infectious agent came from a single source. Now, I guess every disease has to come from a single source at some point, right? AIDS has been around since my lifetime. When I was a little kid, there was no such thing as AIDS. Um, at least if it was pretty early on in my lifetime because I remember hearing about it when I was fairly young, but I think the 70s, and maybe it was before I was born. But anyway, it's certainly not been around forever. But at some point, these things are going to move from being a point source type of an infection to the other kinds that we're going to get to a little bit later. But anyway, for these things that are like smallpox, for example, that are pretty much eradicated, or polio in certain in certain countries and things like that, if we get an infection, it's going to be a point source epidemic. Somebody goes, maybe travels to another country, and they get an infection and come back to, say, America and begin infecting people in their city. So all of the victims with a point source epidemic were exposed from that source. They all came into contact with the person who was originally infected. The next kind is called, oh, let me go back to this. So this is the kind of graph that you're going to get. So here's the date, and here's the number of cases. So apparently we have a handful of cases on January 15th. On January 16th, we have 60 cases. And then I guess people begin to get treatment, or the person who gets sick maybe goes and lays in bed, and so all of a sudden we see this drop off in cases. Common source epidemic would be the next type, and this one you can see just follows kind of a gradual curve up and down like this. And these result from a common exposure to a single source of an infection over a period of time, such as a water source, a food source, etc. Like I remember, I think I mentioned this, that a raccoon or something died in a water tank, in a city, and all of a sudden all these people are getting sick from it. And this would be the curve that you would see from it. And then the last type is called a propagated epidemic. And this is an infectious agent that is communicable from person to person, but then is sustained. So probably a less virulent and a less lethal form of an infection compared to the point source infection. So example, influenza. This is the curve that you would see. So you see a slow beginning, and then all of a sudden it just exponentially climbs and then kind of flattens off a little bit. So once epidemiologists are able to gather enough data, they can begin to see which type of a statistical trend these epidemics are following. So this term, the index case, this is the term given to the first person that they find that has the disease. Now, it's not necessarily the first person who had the disease, but it's the first person that made them aware of the disease. Okay. So right now we're going to talk about some words that you've all heard, and we're going to make sure you understand what they are. The first one is called endemic. Okay, so if something is an endemic disease, it has endemic occurrence, it exhibits a steady frequency over a long period of time in a particular geographic locale. So this right here is showing endemic diseases, or an endemic disease near the US-Mexican border here. And I'm not going to venture a guess as to what this is for fear of being wrong or offending somebody, because I have no idea. But what you see with this is you're going to see a steady number of cases in this geographic location pretty much all the time. Another type that we're going to run into is called sporadic. So something that has sporadic occurrence would be occasional cases reported at irregular intervals, just at random locations. Tetanus would probably be a great example of this. The next type is called an epidemic. And that's when either an endemic or a sporadic disease begins to increase beyond what is expected for a normal population. Okay, so time period not defined, can be hours or years. But the biggest thing with epidemic is that it stays within a country or sort of a more geographic location. Okay. A pandemic, oh, whoops, I'm getting a little ahead of myself, sorry. So the exact percentage of increase needed before an outbreak qualified as an epidemic is not defined. So it's sort of like, okay, we've got a certain number of cases, let's take a vote. And is this considered to be an epidemic or not? You know, all those in favor, yes. All those not, no. So it's not clear at what point it actually becomes an epidemic. But the point is, epidemics are a lot more than what you would see with an endemic or sporadic occurrence. So let's talk about that. So let's say you have an endemic disease, and then all of a sudden, more and more people are all of a sudden getting infected with the same disease. What might be an example or a cause of that? Well, potentially it could be something like a mutated strain of the disease that is more virulent. Okay, so now pandemic. So a pandemic is when you get an epidemic that goes basically worldwide. That's what pan means. So a worldwide epidemic is a pandemic. So spongiform encephalopathy would be an epidemic in Britain. Never made it to the U.S. Had it made it to the U.S., it would have been an epidemic, right? Or excuse me, a pandemic. Swine flu. Not sure if it ever got considered to be pandemic, but certainly it did cross borders. Same with the avian flu. Most of these are just in movies. Which of the following terms describes the total number of persons afflicted with a particular infectious disease in an entire population? Morbidity, prevalence, mortality, or incidence, or endemic. And so this is afflicted, not necessarily dead, right? And so the answer is prevalence. So it wouldn't be incidence because incidence is the number of new cases over a period of time, not the total number of cases. And it wouldn't be morbidity because I'm not actually sure, because you could certainly argue that this could be morbidity. Very, very similar. So anyway, that is chapter 11. Hopefully that made sense. Please study hard, and we'll move on to chapter 12.